Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT class 12th molecular basis of inheritance, that is part 8, transcription. What is a transcription? The process of copying genetic information from one strand of the DNA into RNA, it is called as a transcription process. The transfer of the genetic information from DNA to RNA, that is called as what? They are completely transcription process, means simply called as any DNA to in a RNA. That is called as what? Transcription process. Here, this also in the principles of the complementary groups, the process of the transcription, except the adenosine, the complementary now the forms base pairs, the uracil instead of the thymine. So when you hear what happened, mostly here thymine is present in that RNA. There instead of what? Uracil. They are thymine, they instead of their uracil they get present. However, they are unlike their process of the replication, while the set of the total DNA, their origin get as a duplicates, the transcription only a segment of the DNA, only one of the stands they copy from RNA. So total, for example, this is a DNA stand. In this total DNA information does not to copy into as any in this RNA. Do not completely they get to copy into as any RNA. Only a segment of the some information only they get copy into as any where into the DNA to RNA. Only some information to get copy. Correct. This necessity they are defining the boundaries that would be they determine the region of stand the DNA that would be their transcript. Then what happened? This total information they get transferred here to here, that is called as a transcription process. Okay. Now, why both strands are not copied during the transcription has in their simple answer. So why they are both strands they get involving here? Why they are both strands are not involving? So they have some reasons as present here. Let's see there. The first one, the both strands they get act as a template. So this also they get act as a template. It is also they get act as an in the template. Then what happened would be the code RNA molecule with as a the different sequence. The remember the complementary does not they're identical. It turns into as the protein the complete code for as a protein sequence and amino sequence. The proteins would be there different. So this amino acids and this amino acid proteins amino acids, so protein sequence they completely get be they are mismatched. Then what happened? The complete the transcription it does not takes place. Only one segment will be get participate. What happened? That sequence will they get perfectly they get matched here. Okay. Hence here one of the segment of the DNA would be their code for as two different proteins. So here one protein, here one protein. The two proteins they get participate. Then what happened here? They're mostly the two proteins will be they get at the same time at a time in the DNA will be their bind. What happened to completely the DNA will replication does not takes place, and would be the replicates of the genetic information the transfer in missionary. So information also does not they are carrying. Next, the second, that is an RNA molecule that is an produced as simultaneously will be then complementary to each other. So then what happened? It is also to get produced as information. It is also to get produced as information. So here to get form as the double stranded RNA. Here the double stranded RNA. This would be the prevent the RNA from beginning the translated into as a protein and exercise the transcription become would be their failure once. So the both RNA strand, it is also RNA strand, it is also RNA strand. What happened? The transcription is does not takes place. Okay, this is a information about as a transcription. Okay, let us now discuss the transcription unit. The transcription unit. The transcription unit DNA is defined primarily by the in three regions in the DNA. The mostly the transcription they are entire they're defined primarily in three regions. The first one is the promoter, structural gene, and then terminator. They have in three regions. The first one is in a promoter, structural gene, and then terminator. Correct? Let us see here. The first discuss one by one. Here in this total transcription process, the DNA is the convention of the defining the the strands of the DNA, the structural gene of in a transcription unit. That is, since the two strands there have opposite polarity and the DNA dependent RNA polymerase it also catalyst in the polymerization of only one direction from as in 5 prime to as in 3 prime, and the strand that has in the 3 prime to 5 prime, that is in a template. It is in the template, that is the stand. So for example, here when we take as an A, there one stand. See, this stand there get first here when you take in this total the DNA templates, it is a 5 prime like 3 prime and 3 prime to as an A 5 prime. Am I right? So in these one catalyst they are involving, that catalyst name is what, that is in a the DNA dependent RNA polymerase. Here the DNA dependent, there is an RNA polymerase. RNA, the polymerase enzyme, they are get participate. Now here in this process they get completed, they are following a three steps. One is in a, this is in a promoter region, it is in a promoter and it is completely structural gene, and this process is called as an, they are terminate. This is in a terminator, promoter, structural gene and then terminator process. And after when see here the other stand which be there polarity, the 5 prime to the 3 prime, that is sequence of the RNA that accept the thymine that replaced with as an uracil. If in case for example this is in a, the DNA stand, here compulsory that RNA stand get the instead of thymine they present as what, uracil is present there, instead of the uracil is present. Now the slightly the stands which does not their code there anything, that's referred to as the coding stand. So here in this total process, one for as they get act as in the coding stand, then another one get act as what, in completely template stand. See for example here in this process it is a 3 prime to as an A 5 prime one direction and 5 prime to as an A, they are 3 prime direction. Let's see in these the total in this region they get mostly they get participate in the different the codons they get bind within the stand. Let us see. All the reference the point of view that defining the transcription unit that made with as a coding stand that explain the point of their the hypothetical sequence of the transcription unit, they are represented below. So how to the total the transcription unit that takes place, they given as an A1 enter the coding stand also they given here. That is first here the adenine, thymine, guanine. Already you know that adenine bind with as a thymine and guanine bind with as a cytosine bind with as an adenine. So there is a what, it is in a get act as in this is in a template stand and get it is act as in a, that is in a coding stand. One is in a template and one is in a their coding stand. Here you can write the sequence of the RNA that are transcribed by the about the DNA. So how, it is a very simple one. What is that? In this where the thymine is present in the, instead of they having as what, the uracil they get bind. Here they get bind with as what, the uracil is present here. Am I right? That is, let us after once you that observed, it is in a promoter region, it is a terminator region. Am I right? Let's see. Here the promoter and the terminator. Here promoter and the terminator, the flank of the structural gene. So it is in a structural gene. Am I right? See, this part is called as what? It is in a structural gene, and in this region has a promoter is present. In this region has a the completely terminator region is present. In this flank of this completely the present of what? The present of in a the structural gene is present. Let us after in the transcription unit, the promoter said to be as located towards the end of the upstream. So where the 5 prime the end is present, here they having is a completely the term, the promoter is present, it is in a 5 prime end. That is upstream process. It is called as what? It is an upstream and it is called as a downstream. It is an upstream, it is in a downstream process. And of the structural gene that refers to as made with as in the respect to the polarity, the coding stack. So it is there complete there explaining about as polarity of the coding stack. And here it is a DNA sequence that is in a provides the binding site RNA polymerase. So now here they bind with as what? Here to get bind with as here in a DNA dependent RNA polymerase they get act as in a catalyst now. So here they get start. Let us, and the presence of the that promoter is in a transcription unit, it is also defined as in that in a template, and coding stand. So then after what happened in this transcription unit, this total is called as a transcription unit, right? In this total transcription unit, they get completely, they get explained about as in the template stand and then coding stand. And by the in a switching the position which at the terminator, the definition of the coding template, the start could be there in a reversal. So when this enter the promoter, they get starting, and their terminator will be they get completely end of the region. Then what happened? It is the total the codon. Here what happened? It is a coding stand. It is in a template stand. So when they get reverse, what happened? It is the completely the codon means it comes as a coding sequence. Here comes as in a, that is in a coding sequence, and it is comes as what in a, that is in a template, that is in a, the template stands there comes. And next here the terminator is located towards in a 3 prime the downstream, the coding stand as an usual they defined as the end of the process the transcription. Yes, so before we see it is in the terminator the transcription unit, what happened, it is in it the terminator region is present, that is in a completely the 5 prime, the 3 prime region. Here the additional their regulatory sequence, they may be the presence of as in that upstream downstream the promoter. So here the upstream process, and here they having as in a downstream process, they depending upon the formation of the what in a, that is in a either template stand, either in a completely the coding stand, template stand and coding stand. Since of the any properties of the sequence shall be there describe their their dealing with as in the regulation of the engine expression. So this total information they get completely explained about the process of in what, that is in a gene expression, that is in explained but as what gene expression. Okay, remaining to discuss in next class. Thank you for watching. Please share, subscribe and like.